Welcome to the fourth video in the AP Biology series. So this is the last video of the Unit 1, Chemistry of Life. So in this video, we are going to be going over the macromolecules lipids and nucleic acids. So let's start with lipids. This photo shows a property of lipids. So I want to have you take a look at this and think about what is the property that's being shown in this picture and what's the chemical explanation for that property. So what's being shown here is lipid, so maybe oil, as well as water. And you can see that the oil is forming droplets and really staying away from that water. That is sort of demonstrating the property of lipids, that they are hydrophobic. And the reason they're hydrophobic is that they are nonpolar. Water, you remember, has hydrogen and oxygen, which is a polar bond. So the water molecule as a whole is polar, whereas lipids are nonpolar. So why are lipids nonpolar? What is it about their structure that leads them to have a nonpolar property? Lipids in general are made of a lot of nonpolar bonds. So you'll see that there are a lot of carbon and hydrogen bonds in lipids. Lipids don't have monomers, but they do have building blocks. And so in the other macromolecules that we've seen, there are individual components that form repeating structures within the overall macromolecule, within the big molecule. Lipids don't have that, in the sense that they don't have one single molecule that can continue to add and add and add and add and add and make the macromolecule larger and larger. Lipids, though, do have building blocks. So some really common building blocks that I want you to be familiar with is glycerol, shown at the top there, and then fatty acids. There's one type of lipid that is formed with a backbone of glycerol and then 3 fatty acids. So what's the name of the process that would allow us to build these larger molecules out of smaller building blocks? And then also, what's the process of the larger molecule breaking down into the building blocks, a big molecule becoming smaller? Just like we've seen in the other macromolecules, in lipids, the process of the building blocks coming together to form a larger molecule is called dehydration synthesis. And you can see that moving from the left to the right. Breaking this molecule up is the process of hydrolysis. And just like we've seen before, dehydration synthesis involves an OH and an H leaving the molecule, and that's what forms water. And then that leaves a covalent bond where the OH and H came out. And you can see that here happening 3 different times. So the glycerol molecule on the very left has 3 attachment points, and you can fit other molecules into each of those attachment points. In this case, it's a fatty acid that inserts itself in those glycerol attachment points in all 3 of those locations. And so this is called a triglyceride because there are 3 of them. So what do you notice about fatty acids? Why are they called fatty acids? What are they? So fatty acids are primarily a hydrocarbon chain. You see that carbon and hydrogen. And that's going to be nonpolar. Those carbon-hydrogen bonds, carbon and hydrogen have very similar electronegativities. And so carbon and hydrogen in this type of molecule is going to leave that portion of the molecule as nonpolar. On the very left side of those fatty acids, though, you'll notice a carboxyl group. And you remember that a carboxyl group, as a functional group, has an acidic property. That's where that name acid comes from. So these are fatty acids because they have that hydrocarbon chain as well as the acidic portion. It's that carboxyl group that allows for the connection of the fatty acid to the glycerol. All right, so here we see a couple examples of triglycerides. And remember, that's 3 fatty acids with a glycerol backbone connecting them. What do you notice as a difference between these two, saturated versus unsaturated? What do those terms mean? So what saturated means is that it can hold, or it's holding as many hydrogens as it can. So each of those carbons, if you look at sort of the carbons in the center of that fatty acid chain, each carbon is bonded to 2 other carbons and then 2 hydrogens. In the case of the unsaturated fat, at least 2 of those carbons are double bonded, which means that they're using an extra one of their connection points to bond to one of their neighboring carbon molecules, and that means that they don't have a connection point to bond with the hydrogen. This changes the shape of the molecule, it kind of creates a kink, which means that those molecules as a whole can't get as close together. And so what that means is it's going to stay liquid even at relatively cool temperatures. So like at room temperature, oil, which is unsaturated, will stay liquid, whereas butter, which is saturated, will be a solid. So let's look now at a different kind of lipid, which is a phospholipid. If you remember the triglycerides, there was the glycerol backbone with 3 fatty acids connected to it. In this case, what do you notice? What's a similarity and a difference comparing phospholipids to triglycerides? So if you look at the phospholipid structure, and you can probably see that most easily on the very left side of this diagram, you'll notice there is still a glycerol molecule. And the glycerol molecule is bonded to some fatty acids. But in this case, it's only bonded to 2 fatty acids, which means it has one more connection point available. That final connection point is bonded to a phosphate. And that's what makes this a phospholipid. So phospholipids have the unique property of having I guess it's not that unique, but it's an important property in this case, of having one part of it be nonpolar and one part of it be polar. So those fatty acids, you remember, are made of lots of carbons and hydrogens, so those are nonpolar. Those stay away from water, right? So those are hydrophobic, whereas the phosphate head, the phosphate side, is polar and likes to form hydrogen bonds and get close to water. So you can see that on the very right side of this diagram, that phospholipids tend to form a bilayer, which sort of protects those non-polar fatty acid tails on the inside away from water, whereas all of the phosphate heads are pointed towards the water on both sides. So that's what a cell membrane is, for example, and we'll see phospholipid bilayers in more detail in future units. But you can see that there's water on both sides of that, near the phosphates of both sides. And those fatty acids, those hydrophobic tails, are pointed to the inside, away from the water. Here's another diagram of phospholipids, and here we can see phospholipids within a membrane. So the phospholipids are shown in gray. You can see those phosphate heads and the fatty acid tails. Remember, those fatty acid tails are nonpolar and hydrophobic, so they're going to hide from water, whereas those phosphate heads are polar and so are going to associate with water, both inside and outside of the membrane. You can see there are lots of other molecules embedded within the membrane. And again, we'll get more detail on that in later units. For now, though, I want you to understand the term fluid mosaic model. Think about each of the components of that term and figure out what you think that means. What is a fluid mosaic model? Fluid means that it can move around. Fluids take the shape of whatever container they're in, so things can move around within them. They're not solid. Mosaic means it's something big made up of lots of little things. So the model of a cell membrane is that there is a lot of movement of the pieces within it, and that there are a lot of different pieces within it. So you can see all sorts of things embedded within that phospholipid bilayer, mostly proteins, but there are other components as well, such as cholesterol. And all of those pieces can kind of move throughout the membrane because each of those phospholipids can move around a bit as well. All right, so let's talk about a third type of lipid, which is steroids. So steroids are lipids because they are blank. Think back, what do all lipids have in common? Why are lipids lipids? What do they share as a property? So lipids are hydrophobic. And lipids are hydrophobic, that means they tend to stay away from water, right? That means they're insoluble in water. But steroids don't resemble other lipids since they have a structure composed of 4 fused rings. Earlier we saw examples of lipids that have a glycerol backbone with 3 connection points, and we don't see that here in steroids. One of the most common types of steroids is cholesterol, and cholesterol is the precursor to all sorts of molecules, including vitamin D, testosterone, estrogen, progesterone, aldosterone, cortisol, and bile salts. So here we have a diagram of an example of a steroid. Something I should point out is that all of these steroids, because they are hydrophobic, they are going to be, they have different properties than a lot of other hormones, right? Like testosterone and estrogen are hormones. But these hormones, these are going to be hydrophobic hormones, and that's going to become really important in a later unit. So keep that in mind. All right, here's a moment to review. So we went over 3 types of lipids. What are they? So the 3 types of lipids that we talked about were triglycerides, phospholipids, and steroids. And you can see diagrams here showing their component parts. So next up, we're going to go on to our last macromolecule, which is nucleic acids. So to start talking about nucleic acids, let's talk about the monomers of nucleic acids, which is a nucleotide. What are the components within a nucleotide? And I want you to try drawing a generalized nucleotide of DNA, and then within the sugar portion, and that's a clue, there is a sugar portion, within the sugar portion, number the carbons on the sugar starting from the base. This might have been a hard one to draw all of those components, and that's okay. What I really wanted you to focus on was the 3 subcomponents. It's okay if you didn't get the details of what, for example, the nitrogenous base actually looks like. But you should know that a nucleotide has a phosphate group connected to a 5-carbon sugar, otherwise known as a pentose sugar, and that's connected to a nitrogenous base. The reason I asked you to number those carbons on the sugar is that's going to be really important as we go into more detail of studying DNA. Knowing where especially that 3rd carbon is and the 5th carbon is really important. So if you notice, it's the 5th carbon on the opposite side from the nitrogenous base that's bonded to the phosphate group. So keep in mind the 5th carbon of that pentose sugar is bonded to the phosphate group. Nucleic acids include both DNA and RNA, so now's a chance to see what you remember about what the differences are between DNA and RNA. So the differences between DNA versus RNA that can be seen here, let's start from the sugar itself. DNA is named DNA for deoxyribose. So the sugar of the nucleotides of DNA is called deoxyribose, whereas in RNA it's called ribose. Why do you think the DNA is called, why is that sugar called deoxyribose in DNA? Why is it, well, I guess not why is it called ribose, but why is it called deoxyribose? So it's called deoxyribose because compared to ribose, we've lost an oxygen. So it's deoxy, that oxygen is taken out. All right, another difference between these is the bases, the nucleobases of DNA versus RNA. In both cases, we have 4 nucleobases, and that's the nitrogenous base, but the 4 bases are not identical. I'm going to read from the bottom up. In both of them, DNA and RNA, we have guanine. We also have adenine in both of them, and we have cytosine in both of them. But that 4th base is different in DNA. In DNA, we have thymine, whereas in RNA, we have uracil. Another difference, and it's not always true, but is often true, is DNA is usually, but not always, double-stranded, and RNA is usually, but not always, single-stranded. So you can see that in this diagram. So I have one more question for you before we go on to the next slide, which is a bit of a review. What are the names of the monomers of nucleic acids, and how are those monomers connected to become a polymer? So the monomers of nucleic acids are called nucleotides. That's what we were drawing at the beginning of this section. And just like in all the other macromolecules, the process that brings those monomers together and connects them with a covalent bond is dehydration synthesis. And the opposite process is hydrolysis. So once again, we can see dehydration synthesis moving from left to right in this diagram, where we're losing an OH and an H, and that what is left over at the end of that process is a covalent bond. So we have 2 molecules, 2 nucleotides on the left side, and then we have 1 molecule on the right side, because those are now connected with a covalent bond. So this diagram is showing how the nucleotides are connected to each other along one strand. Let's imagine we have DNA, and remember, DNA is usually double-stranded, right? So here it's the dehydration synthesis that is causing the nucleotides to connect to one another to form one of those strands. What connects the 2 strands to each other? So the two strands are connected together by hydrogen bonds. Now remember, hydrogen bonds is much weaker. It's not a covalent bond, so this doesn't create one molecule, right? When we have things hydrogen bonded together, they're still two different molecules. So technically, the two strands of DNA are still separate molecules, but they are connected fairly stably with those hydrogen bonds. A great thing about that hydrogen bond connection being not quite as strong is that we can kind of open it up and then close it up. So that allows us, when we want to get information from the DNA or to copy the entire strand, we can just sort of open up those hydrogen bonds and get at the information in the middle and then seal it up again. All right, so not every base can connect to every other base. What bases of DNA connect to each other? So we can actually see the answer to that question in this diagram. We can see that thymine and adenine bond together, and guanine and cytosine bond together. A couple things I want to point out here. One of the reasons these pairs bond is due to the number of hydrogen bonds. Thymine and adenine have 2 spots where they can connect, and guanine and cytosine have 3 spots where they can connect. Another thing I'd like you to notice is the number of rings in these bases. Notice that thymine has just 1 ring, where adenine has 2 rings. Guanine has 2, cytosine has 1, which means that each of these pairs, like the AT pair and the CG pair, have a total of 3 rings going across. That keeps the structure the same width across all of these pairs. The next question I have for you about the structure of DNA is, what does it mean that there is an anti-parallel structure? So anti-parallel means that the nucleotides of one strand of DNA are running in the opposite direction to the strands, or to the nucleotides in the opposite strand of DNA. So if you notice on the left side of if you look at the diagram where you can see the nucleotides themselves, you'll notice on the left it says 5' at the top, and on the right it says 3' at the top. And what this is telling you is which sugar is sort of facing in that direction. Remember that the carbons in that sugar all get numbers. So remember that the 5 carbon is bonded to the phosphate. And so on sort of that left strand, again, I'm looking at the more blown up version here. So the 5', you can see it's the phosphate, then the sugar, then the phosphate, then the sugar. Whereas over on the right side, on the very right side of this slide, you see sugar, phosphate, sugar, phosphate. So these nucleotides are kind of reversed to each other. One's running one way and the other's running the other way. That's going to be really important to keep in mind as we study DNA in more detail. All right, final slide. I want you to take a look at the molecule pictured here. Try to figure out what it is. So hopefully you noticed that there's a base, there's a sugar, and then there's a phosphate region. And that base sugar phosphate means that it's a nucleotide. This nucleotide looks slightly different than other nucleotides we've seen, though, because here there are 3 phosphates. So nucleotides have only 1 phosphate when they are bound in DNA and RNA. However, nucleotides can technically have 1, 2, or 3 phosphate groups attached to them. And free-floating nucleotides often have additional phosphates that then will be removed as a part of that combination into DNA. So the removal of additional phosphates provides the energy for reactions. So this includes the synthesis of DNA and RNA. Sometimes, though, that energy is providing energy for different kinds of reactions, not just the synthesis of DNA and RNA. This molecule is actually a molecule of ATP. ATP stands for adenosine triphosphate. So that base pictured here, and you don't need to recognize it at this level of detail, but that base is adenine. And so when you have adenine with 3 phosphates, that's adenosine triphosphate, or ATP, which is going to be incredibly important to us in studying biology this year. So this is a great molecule to get to know. So this is our final slide of the video today, and that also means this is the final slide of this unit. So we are finished up with Unit 1, Chemistry of Life. So congratulations, you've reached the end of Unit 1.